All right, let's jump right into this masterclass explainer on mitochondrial transplantation therapy. Today, we're taking the incredibly detailed material from lectures 10 and 11 and translating it into a dynamic, start-to-finish guide. We're going to cover every crucial mechanism, experimental approach, and translational challenge so we can really understand how scientists are literally learning to hotwire human cells. So, here's our roadmap for today. We're starting with basic cellular bioenergetics, moving step by step through cutting-edge cell therapy applications, exploring specific disease models, and finishing up with a high-yield exam prep session. Okay, Section 1, Mitochondria, Structure, and Function. Let's build a strong foundation on what these incredible organelles actually do. To really grasp this therapy, we absolutely have to master the biology first. Think of oxidative phosphorylation as a city's power grid. It's the process where the electron transport chain generates ATP, our fundamental cellular energy currency. Now, uniquely, mitochondria contain their own separate genetic blueprint, mtDNA. These 37 crucial genes encode 13 essential respiratory proteins. But here's the catch, and it's a big one. Unlike our nuclear DNA, mtDNA is highly vulnerable because its repair mechanisms are super limited. If those specific proteins mutate, well, the entire power grid just shuts down. And when we actually look at these organelles under a confocal microscope, we see they aren't just floating around like static little beans. No, they form these highly dynamic, glowing, interconnected networks. They're constantly moving, dividing, and fusing to meet the cell's energy demands in real time. It's really something to see. So it's definitely time to shatter that old textbook definition of mitochondria as just the powerhouse of the cell. I mean, yes, they make energy, but they're actually master regulators. They actively dictate our immune responses, they set our circadian rhythms, maintain calcium homeostasis, and even control gene expression through epigenetic pathways. Because they're integrated into almost everything we do, a failure here causes catastrophic ripple effects across the entire body. Moving on to Section 2, Dysfunction and Disease Pathology. What actually happens when this intricate power grid begins to fail? Mitochondrial dysfunction is essentially a perfect storm driven by genetic mutations, aging, and even just sedentary lifestyles. When these factors compound, they drastically increase reactive oxygen species, or ROS. Think of ROS like toxic exhaust fumes. While a tiny amount is totally normal and used for cellular signaling, an excess completely destroys mitochondrial structure, specifically targeting essential lipids like cardiolipin. And that disrupts the critical balance of fission and fusion, creating this vicious cycle of structural collapse, rampant oxidative stress, and ultimately plummeting energy output. Now, this brings us to a highly testable concept that explains why genetic mitochondrial diseases are so clinically unpredictable. It's called heteroplasmy. Basically, this is the biological coexistence of both mutant and healthy mitochondrial DNA within the exact same cell. A disease phenotype, like renal failure, myopathy, or seizures, only manifests when the ratio of mutant to healthy mitochondria crosses a critical threshold. Stay below the threshold, and the patient appears perfectly normal. Cross it, and the whole system crashes. Section 3, the promise of MTT. With over 300 different diseases linked to this dysfunction, let's explore how mitochondrial transplantation therapy offers a literal biological rescue mission. Right now, modern medicine is essentially fighting these diseases with one hand tied behind its back. We really only manage symptoms using basic supplements like CoQ10. There are zero cures for genetic MTD disorders. So MMT represents a massive paradigm shift. Instead of just slapping a chemical Band-Aid on it, we're attempting direct organelle replacement. We're delivering fresh, healthy mitochondria into compromised cells to restore bioenergetics and stop cell death at its actual source. And listen, this isn't science fiction anymore. It's a rapidly accelerating reality. The evolution of this science is fascinating. It started way back in 1982 with some early observations in cancer cells. By 2009, researchers achieved groundbreaking physical transplants in rabbit hearts. Fast forward to 2024, and we've successfully completed phase 1 human trials for ischemic stroke with no adverse events. We now have the very first evidence that brain mitochondrial transplantation is actually safe in humans. Section 4, Disease, Models, and Applications. Let's look at the hard clinical data to see how all this translates into practical use. Across severe genetic models, the experimental success is honestly staggering. Take Lee's syndrome, for example. Injecting wild-type liver mitochondria into knockout mice, actually improved systemic energy metabolism and prolonged survival. For MELAS, researchers used mesenchymal stem cells, MSCs, to deliver mitochondria via endocytosis. They literally had the cells engulf the organelles, restoring cellular respiration for weeks. And for MELRF and LON, scientists are actively engineering advanced peptides, like PEP1, and using nanoparticles to safely escort the organelles right through the cell membrane to successfully reboot ATP production. But the applications extend way beyond inherited genetics, directly into severe neurological trauma. In Parkinson's disease models, introducing healthy mitochondria drastically reduces microglial inflammation and helps restore dopaminergic neurons. In spinal cord injuries, local mitochondrial transplantation actually prevents demyelination and drives genuine functional recovery. Why does this matter? Because we're essentially handing damaged nervous tissue a massive, targeted energy battery so it can actively heal itself rather than just slowing down the damage.
Section 5. Critical translational challenges. As you can guess, translating these spectacular lab successes to the bedside comes with some objective roadblocks.
The lecture identifies 5 major translational hurdles we've got to clear. Number 1, viability. Isolated mitochondria are incredibly fragile, surviving only about 1 to 2 hours on ice. 2, the immune response. Using donor mitochondria can trigger severe pro-inflammatory cytokine storms. 3, targeting efficiency. Currently, only about 10% of systemically delivered mitochondria actually reach the target tissue. 4, genomic compatibility. Mismatches between donor mtDNA and recipient nuclear DNA can disrupt cellular harmony over time. And finally, 5, scalability. Time-intensive isolation just doesn't work for fast-paced emergency medicine. But bioengineers are fighting back with some truly brilliant solutions. To solve viability and targeting, researchers are encapsulating mitochondria in engineered vesicles to safely smuggle them right across the blood-brain barrier. To bypass immune and genomic mismatches, they're synthesizing iPSC-derived mitochondria, creating perfect autologous matches from stem cells. And incredibly, they're even combining MTT with CRISPR technology to precisely snip out pathogenic mutations before the transplant even happens. It's next-level stuff. Okay, Section 6, lecture recap and exam prep. For those of you studying this material, let's lock in these high-yield concepts with some specific exam questions pulled directly from the lectures. Here's a classic fundamental exam prompt to test your grasp of the core mechanism. Why can't we just use a patient's own mitochondria, an autologous source, to treat their congenital mitochondrial disorder, thereby bypassing the immune system entirely? The answer is straightforward but absolutely critical to memorize. If the disease is congenital, the patient's entire systemic power grid is fundamentally flawed. Their inherent mtDNA carries the mutation in every single cell. Harvesting and replanting their own defective mitochondria achieves exactly nothing. You have to use a healthy, external allogeneic donor to introduce functional, wild-type DNA into the system. Next question, and I guarantee this is going to show up when discussing pathology. Can you precisely define heteroplasmy and explain exactly why it makes the clinical presentation of these diseases so varied? So, heteroplasmy is the biological coexistence of both mutant and healthy wild-type mitochondrial DNA within a single cell. Its role in disease expression acts exactly like a biological tipping point. A patient won't show any physical disease phenotype until the proportion of mutated DNA accumulates, crosses a critical threshold, and just completely overwhelms the healthy mitochondria's ability to compensate. Looking ahead, the frontier of this science is truly staggering. We're witnessing the real-time convergence of targeted nanomedicine, precision cell therapy, and genetic engineering, all poised to completely rewrite how we approach cellular energy and treat once fatal conditions. I'll leave you with this final powerful thought drawn directly from the lecture's conclusion. We are literally learning to hotwire human cells. By replacing the engines of our biology, we're turning the untreatable into the curable. With phase 1 trials already proving safety in the human brain, the question isn't if this therapy will work, but rather, what barrier will cellular energy medicine break next?